Martin Borch Jensen, who is the Chief Scientific Officer and co-founder of Gordian Biotech, is going to be talking about the biology of aging 101, right? So it's the foundation for this week. He gives an incredible lecture on this subject. A PhD at the National Institute of Aging at University of Copenhagen, postdoctoral research at the Buck, who will be visiting on Friday, really the leading institution for longevity here. And then left there to go start Gordian. He's also founded Norn Group. It's a nonprofit focused on accelerating aging. Like me, he's surrounding aging from a multitude of different directions. A fun fact, he published a book on intermittent fasting while completing service in the Danish Air Force. And do you still intermittently fast?
I don't currently, and I'm going to talk about that.
Yeah, so it's an interesting, right? There is a debate about whether you should or shouldn't. Some of the actual science, unless you're overweight, is not necessarily proving it out to be beneficial, especially if you're looking to add muscle mass. So super excited about that. And then we're going to be having you join us for Q&A at lunch. And then we'll be actually diving into Gordian Bio through his partner right afterwards, which is, I think of Gordian Bio as the mixture of AI, single cell sequencing, and trying to make incredible discoveries. Thank you for joining us. A pleasure.
Thank you, Peter. And thanks everyone for being here. I think it's, obviously, I think it's a very...
Before we start, let's give it up for Martin.
Yeah, so I was saying, obviously, I think this is a tremendously important field. The reason I'm in longevity, when I was 16 or something, I was having my existential phase and trying to figure out the meaning of life. I don't think I figured it out, but I came to the interim conclusion that being alive is required for whatever the meaning of life is, and that's what we should focus on. And then I've been sort of exploring my way forward to what do we actually do. And then along the way, sort of realizing that even if, whether or not you're motivated by extending lifespan, we all want health. And if most of the things that plague us are things that sort of happen with age, then the possibility of regulating the underlying mechanics of that is the biggest lever we have on health in this country or in the world generally. And so whatever your motivation, I think that's just a tremendously sort of like important and underappreciated field. I'll skip over most of the introduction since Peter covered it. I'll just share who am I according to NIH when they're reviewing my grants. I'm apparently a hardcore scientist. I still think that that's true. And then, timeline as Peter went on. I've been studying aging since before my PhD, wrote a book on intermittent fasting. It's in Danish only, but anyway, it's out of date now. While I was in the Air Force, did more research at the Buck Institute, then founded Gordian with Francisco, whom you'll hear from later. And then I have this nonprofit Norn Group. There's multiple programs there. There's a big academic grants program called Impetus Grants, and there's some training programs and so forth. Basically, all of it is like, how do we get to serious results in the next 30 years or so? So that's me. This is broadly what we'll cover. Brief, since people are already motivated, sort of intro on like, what are we talking about here? Then I'll spend some time just defining our terms, like what do we mean by aging? I think there's a lot of areas of the aging field where terminology is used sometimes a bit loosely. There are some terms like senescent or inflammaging that are used without a clear definition, and that can lead to some confusion. So we'll spend a bit of time there. Then the bulk of this will be going through different areas of biology that are applicable to sort of the aging process overall. And then at the end, we'll talk a bit about what is the current state of the field, what's going on, what are the major challenges, like what's needed to accelerate progress. So aging. I think you guys have seen a graph that looks something like this before. But it's not just that like you have a linearly accumulating risk of disease as time goes on. It really is an exponential growth in most cases. These are just some indications. Obviously, the financial burden, this is just US costs and it's outdated by some years. is tremendous. That's the direct cost. The indirect cost of sort of lost labor and so forth is not included. So whether you're coming from a sort of like national economy or a personal health perspective, aging is important. And I don't need to remind everyone here, but like you are on the way to the right. And I think that's why you're all here. So, if anyone in the audience felt that you are not affected, that's not true. So when we think about risk factors for disease, I just said that aging is the major risk factor for most diseases. But most of the time, you'll see some overview like this of like, okay, you want to avoid cancer. It's important that you don't smoke. These other things are important. That's true. But the reality is something like this. The dominant risk factor for getting cancer is your age. Why don't we talk about that? And I think the reason is that we also don't talk about luck as an important, factor in whether you're going to get cancer. Mostly, the medical establishment does not think that we can do anything about the aging part, and so there's no point in focusing on it, right? So if that was true, if there's nothing we can do, yeah, that makes sense. We just have to accept more cancer as we age. Of course, that might not be true. But for most of the top 10 causes of death in the US, this is the case, that aging is the number one risk factor. For diabetes, it would be number two. And therefore, if we can do something about it, we can have a huge impact on health. Despite that, this is sort of the funding landscape from the NIH for, they have different institutes focused on different organs. They have one focused on aging. These numbers are from 2017, so everything is sort of 10-15% higher maybe, but roughly the same proportions. The Aging Institute is a lot lower than even one individual disease of aging. And half of the budget for the Aging Institute is earmarked for Alzheimer's research, and so not for the specific mechanisms that underlie multiple diseases. Beyond that, there's a bunch of social studies and so forth. The grand total amount that the US government dedicates to like studying aging biology is about $400 million a year, which of course is a good sum. But it's a lot smaller than even, it's more than 10 times smaller than the Cancer Institute. It's much smaller than the Cancer Moonshot and so forth. So we don't have a ton of funding for aging as a process. Why is that? Should we increase that? That seems like, given the graphs we saw earlier, it seems like that makes sense. But of course, it only makes sense if there is the possibility of like a fruitful outcome here. And I think certainly it's true for the aging field that for the last, 5,000 years or more that it has existed, it has mostly been empty promises. The first emperor of China employed a lot of people to achieve immortality and did not get good outcomes from that. He was told to drink quicksilver and it did not end well. So it only makes sense for us to sort of motivate tremendous investment in aging if there is stuff that can be done. Now, in the last roughly 30 years, there's a lot of new data. Before that, the aging field was people sort of, you can read, if you have the same sort of idea of fun as me, you can go back and read like conference proceedings from 1950s. And people are discussing, is aging a thing that happens in a cell? How are cells related to aging? It was really completely nascent to even think about. But then we fast forward to sort of the 1990s. In 1991 to 93, Cynthia Kenyon and Gary Johnson's labs both found mutations in worms that could double the lifespan and double the healthy lifespan of a worm. So what you're looking at here is a survival curve. The y-axis is how many of the animals are alive over a period of time. We'll look at a lot of those. So this is for the DAF2 mutation, which is in the insulin signaling pathway. I noticed there were no carbs at breakfast. This is potentially why. It's in the pathway that cells use to sense nutrients, and we'll get back to sort of nutrient sensing later. But this shows that the aging process is not some sort of thermodynamic law that just happens to all living organisms. You can change one gene in an organism, and then the biological programs that are running run differently in a way that leads to a lot more health. So this is early in worms. We can look at this from different angles as well. If you just look at the diversity of life, you have different organisms that are similarly sized. They're all mammals. They have very similar biology, but their lifespans differ greatly. And so if you're a mouse and you're comparing yourself to a naked mole rat, we don't have that experience as humans in sort of reality, but it's sort of, I think of it as like similar to if you were in a Tolkien, novel and you're a human, and here's this elf, and the elf just lives 10 times longer than you. Visually, maybe the elves are a bit different than the mole rats, but you have another thing next to you that seems very similar, but can live way longer. We don't have that experience because humans are very long-lived, but that is the reality in nature, and there's no reason to believe that this can't be sort of modified. And then finally, as Peter already pointed out, one cell in the human body has the capability to create a completely young new human body, right? This doesn't happen as sort of like an internal rejuvenation process. It happens as children. But it just proves to us that the biological programming is there to create more cells, create more young cells. So all of this together just tells us aging is a biologically malleable process, and we have made progress in understanding some of that only in the last 30-ish years. Before that, we really had absolutely no clue how any of this stuff worked. And so we'll go through a lot of that biology. Let's get a bit more tangible on how do we think about aging. We can all recognize it, except for with you, right? We all misjudge you as being 60. We can all recognize it, but that's not the same thing as being able to measure it, being able to define it, and so forth. The way I suggest thinking about it as a high level is we have sort of aging as a whole, aging of an organism, and that's the part that we can see, and that's the part that we can feel, like do we feel energetic and so forth. And then on the one side, we have different biological mechanisms that happen inside of your body, which you don't monitor directly, but which is what's studied at the Buck Institute and all the other places that are doing experiments here. And then we have consequences of aging. And so when you go to sort of Congress or the NIH, aging there might mean Alzheimer's, right? That's why they earmarked all this funding for Alzheimer's. But that's really a downstream consequence of the processes that are happening. So if we go back to the diagram we had before, we've got our x-axis, which is sort of our time and our natural aging that's happening. And then we have incidents of a bunch of bad stuff happening on the y-axis. And the differential there, the thing that's driving the fact that you don't get better with age, physiologically, but you get worse with age, that's all the mechanisms of aging. So these are three things that are referred to as aging by different groups of people, and it's important to sort of be clear about which one are we talking about. In terms of the mechanisms, like how does the field think about what aging is? In the last, I guess, century and also millennium, there are a lot of different early theories of aging. Here I'm showing sort of the free radical theory of aging, which was that you have these chemical structures in your body that are very reactive. And so you get chemical bonds happening for a lot of the different cellular components. And that's bad because now they can't do their job. And so you get damage. You get damaged ROS, damaged organelles, and maybe that's how everything is driven. There's this one process that's happening and making things work, and that's driving all of aging. So there are different versions of this, what I call single cause theory of aging, like this is the one thing. And that's mostly gone now. I think at this point, the field definitely recognizes that there are multiple processes going wrong and that they are connected to each other, indicated here by these gears that I guess would never run. But there's multiple things going on, and these are somehow connected, and we should think about multiple sort of different areas of biology when we think about aging. The most well-known sort of codification of this idea that there's a bunch of different processes is a paper originally from 2013 called The Hallmarks of Aging, and it was updated last year, inspired by another paper called The Hallmarks of Cancer that sort of defined this set of criteria to, like, these are necessary and sufficient for a cell to be an aggressive cancer. And so the aging field sort of followed suit and created this list of hallmarks, originally 9, now 12. And I think that was good and bad. It's good in the sense that there was no real sort of paradigm, there was no unifying force for the field at the time. And it's bad because it has come to be taken as sort of, this is aging. Like, these factors are how aging works. And that's not at all the case, even when you read the original paper. It's really a list of topics that researchers who consider themselves to be in the aging field are focused on, and where there are studies being done. These different hallmarks are not at the same level of abstraction. You know, there are some things that are very concrete, like your telomeres getting shorter. There are other things that are describe sort of all of life broadly, like intercellular communication, right? Like, that's not a thing you can go measure. It's not a single thing, right? And so it's a list of topics that you might be interested in, that you might want to focus, but we should not imagine that if we just address each of these things individually, like, we're done and like aging is done. I think that would be not insufficient humility in terms of like how much do we understand. The way that I think about sort of aging is more in terms of coordination, and as an organization that's trying to achieve a specific purpose, which is, okay, we are alive, we are mobile, we are thinking, we are reproductive, and there's a lot of components involved in trying to make that happen. And so if you go read papers, and even in this talk, you'll hear a lot about, you know, this process and this pathway and this thing is going on inside of the cell. And maybe, again, if you're a nerd like me, you'll hear about sort of evolutionary theories of aging. Aging happens because there is not selective pressure post-reproduction to keep an organism alive. And so you have selection's shadow, and that's why everything goes wrong. And those can both be true at sort of different levels of detail. But I think the thing that's often not overlooked perhaps, but not given sufficient attention is that, as I said, staying alive is really a question of different parts of the body doing specific things in appropriate coordination with each other, right? Like, it's not about this thing is good or this thing is bad. And we'll get back to this theme very often, but like, your heart beating faster is not like just a good thing, right? Like, it's not useful if it's not coordinated with, you know, like, how much are you breathing and all these other things. Your heart beating slower is also not necessarily good, right? Like, resting heart rates can be good, but we should think of our bodies as this system of specialized parts where all the parts need to function, and the parts can dysfunction. And a dysfunction is defined as, like, they are not acting in a way that is appropriate to how your environment is and how the rest of your body is working. And in my head, maybe this is because I'm an entrepreneur, but like, in my head, I go back to, like, this is exactly how a company works, or at least let's say roughly how a company works, right? Like, you have different teams and different departments that are doing different things. And you can have individual failures. You can have one employee who is not able to do whatever they're supposed to do, or who's, you know, angry and toxic, or whatever it is. And you can have a whole department that's not really functioning correctly, right? And that will propagate dysfunction to the rest of the organization, where other entities will try to sort of like cover for, or, you know, like, function in spite of this dysfunction. And that we see that in physiology as well, right? When you have a liver that's not performing appropriately and you have different circulating factors in your blood, then the rest of your body tries to compensate and succeeds for some time. So there is importance in those individual dysfunctions and the specific things that go wrong, but it is also critical that the pieces work together. And the way that we should think about improving our age states and sort of doing something healthy is not just we need more of this thing, we need more of this pathway, right? Like if you have some sort of inability to deliver on contracts, simply increasing the size of the sales team is not necessarily a solution depending on how the system is working. We'll get back to this in the end. It makes things very complicated because, like how do you solve something like that? And everyone in this room has extensive experience with this, but I do think it is important to sort of keep a mindset like this and not walk away with like, here's this one thing, and I just need to find the drug that does more of this one thing, and then we're good. Okay, let's talk biology. So I'll go through different levels of organization that fail with age. We have sort of the tissue level, we have the level of cells, and then we have communication between the cells and sort of their coordination. And then in each case, I'll go through what I think sort of curated, these are the main things that you should be thinking about. There's certainly stuff that's left out. You're getting the abbreviated version of this 3-hour talk, and even the 3-hour version can't cover everything. But we have an hour for Q&A later. So anything that you're interested in that I missed, ask about it later. So at the tissue level, we'll go through 3 different types of failure. One is that we lose cells. Another one is that the cells misbehave. And then thirdly, around the cells, the sort of environment of the cell gets worse. And then we'll get to what happens to cause these tissue errors afterwards. So this is 2 pictures of 2 thymuses. The thymus is an organ where your immune cells are sort of educated. They're taught, like what should you attack, what should you not attack. And the thymus undergoes something called thymic involution, where the functional cells, which are sort of stained purplish here, are replaced by fat. And so you see in this newborn, the thymus is composed of cells. And in this 24-year-old, the thymus is sort of 50% replaced by fat. And most of the people in this room, myself included, have mostly no thymus left. Like this organ has just involuted and disappeared functionally from your body. The thymus is an extreme example, but there are other organs that undergo sort of similar loss of cellularity, reproductive organs, obviously, but even your muscle. Here's some images of muscle at a young age, and then you see an increasing amount of sort of replacement of muscle cells with fat cells with age. Exercise helps. So the picture on the far left is a very physically active 66-year-old. So it definitely helps, but it's not enough, right? And I think everyone in this room knows that, right? You can impact your rate of aging, and we don't yet have a way to win that battle. Just sort of hang on. So this happens in a lot of different tissues, and obviously this reduces the productive function of the tissue. Another thing that happens in your tissues is that some of the cells stop being productive members of the team, if you will. And so you're obviously familiar with cancer. And the way that cancer progresses is that you constantly have mutations happening in your cell. And the hallmarks of cancer, which actually are sort of necessary and sufficient lists, say, okay, this has to happen. You have to get the ability to elongate your telomeres. You have to get the ability to ignore slow down signals and immortalize the cell. And so these smaller scale mutations are happening constantly. Most of them don't result in cancer. Even the ones that do, for the most part, you have mechanisms in your body that prevent cancers from becoming malignant tumors. You have an immune system that kills a bunch of these cells that are growing excessively. And you have these different mechanisms that shut the division of cells off. And so in our younger years, those mechanisms work well, and most of these individual events that could turn into a malignant tumor are stopped. And then as we age, those mechanisms work less well, and you have more and more mutations. And so you will accumulate cancers, and your cancer risk is elevated 1,000-fold, as we saw. That's one type of dysfunctioning cell. Another prominent in the sort of aging topic is what's called senescent cells. And so you have a sort of somewhat parallel process going on where there are different signals that cells in your body can receive. Some of these come from sort of external chemicals that are either carcinogenic or otherwise toxic, but also things like injuries where suddenly your cells are dividing a lot in order to recover the structure of the tissue after an insult. And so you have these different signals that in some cases create what's called a senescent cell. And the state of a senescent cell, it's sort of the opposite of a stem cell. If a stem cell is like, cool, I'm young, I can be anything. A senescent cell is kind of like grumpy Clint Eastwood, with a shotgun on his lawn, like, get out of here. I'm not going to contribute usefully, but I'm sitting here and being grumpy. That is, of course, a gross oversimplification. There are biological processes that rely on signals from senescent cells, but especially as they accumulate, the stuff that they secrete, which involves a bunch of sort of tissue remodeling and pro-inflammatory factors, can create a local environment where bad stuff happens. For example, even though senescent cells are thought, one of the ways that you can go senescent is if you have really strong signals to divide and grow and become a tumor potentially, the cell can sometimes shut down and become a senescent cell. However, sort of paradoxically, the presence of a lot of senescent cells in a tissue increases the risk of tumors growing from the stuff that they're secreting. So you have this defense mechanism, but as you keep pushing it, it ends up causing sort of detrimental states in the tissue. These are two prominent cases. There are other ways that cells can misfunction, of course, but the point is that with age, your tissue becomes this sort of heterogeneous mosaic of different cells that are doing different things, instead of a bunch of really aligned cells that are trying to do the same thing. Finally, the environment around the cells also ages, and in many cases, this results in what's called fibrosis, which is sort of scar tissue that's being secreted by different cells. You can see here in 3 different organs. At the top is an actual histology image, and then schematics below, showing more and more of this blue fibrotic mass, which prevents function in the tissue in different ways. In the kidney and liver, you have basically a mass that separates the functional cells, the podocytes and the hepatocytes, from the blood vessels that they're supposed to be filtering stuff out of, and so forth. In the heart, you have decreased coherence of the cardiomyocytes that are supposed to beat together. Where this fibrosis comes from, it includes, for example, the presence of senescent cells triggering sort of an chronically inflamed state, and we'll get back to some of that. But the point is that it's not all about cells. It is also about like the structure that cells live in. There are changes there that happen with age and at the sort of tissue level that often results in fibrosis. And fibrosis is something that we don't have good medical treatments for yet, but a lot of companies, including ours, are working on that. This is a, if you want to go deeper on this topic, slide, which shows the cover of a book by Uri Alon, who's a professor at the Weizmann Institute, called Systems Medicine. I think this is the best overview of why do the different organs in our body fail in different ways. Why, for example, is type 1 diabetes a common thing where you get an autoimmune attack on a certain glandular tissue, but we don't have autoimmune attacks on neurons or on hepatocytes. And so the overall framework is sort of depending on how much a tissue is exposed to external stressors and how many cells are in a tissue, you tend to have different categories of diseases indicated here by the different colors. So you have autoimmune disease in tissues that are highly secretory and have sort of a certain amount of turnover versus tissues where you keep your cells around forever, like your brain. They tend to be afflicted more with degenerative diseases where you gradually lose cells. So the first of the 3 topics that we just covered. So yeah, I think there is only inklings of a framework for like why does our body fail in the way that it does. But I think for any scientist or MD or curious person, this is a great a great book to think about the high level. We have a question. I don't know if I'm supposed to do questions now or after, but let's do it. Systems medicine. And
I think if there's a burning question or a clarification, just raise your hand and pop it in. We'll go in depth, but I want to make sure that we don't miss something right in that moment.
Cool. And his first name is Uri. Yuri. All right, let's move on to cells, and I'll cover some of the major sort of things that are happening within cells and the ways that those fail with aging. We'll start with the genome, then we'll talk about junk, for lack of a better word, and how the cell sort of cleans these things up. And then we'll talk about metabolism, which is obviously very important. It's sort of it's necessary for life, but also is the biggest area of like we've actually found something that changes the rate of aging. So DNA damage. There are a number of diseases with symptoms that resemble aging. They're called progerias or accelerated aging disorders. This is Cockayne syndrome, which is one. This child is, I think, 17 years old, but obviously looks older. And a lot of these diseases are caused by specific genetic mutations in genes whose primary role is to maintain the integrity of our genomes. And so that's sort of notable observation that there are at least 6 or a dozen different diseases where you're supposed to have DNA repair and you don't, and then this happens. It's also true in model organisms. These are mice where mutations are specifically introduced in mitochondrial DNA rather than our sort of nuclear genomes. So the rate of mutation is increased 1,000x compared to what normal animals experience. And the animals, you can see on the left here, the bottom is a normal mouse, and then the top 3 are these mutant polymerase gamma mutator mice. And they have different degrees of kyphosis, which is sort of the spine misalignment and graying of hair and so forth. And you can see on the right that the lifespan of these mice is greatly shortened. And so in general, when we don't have our functional DNA repair, animals will live shorter, patients will have phenotypes of aging. This is a recent paper out in Nature that looked at the rate of accumulation of mutations on the left. They got samples from a lot of different species and then measured how fast do mutations accumulate in the species. It was primarily focused on the intestine, but in some cases, at least, this holds in other organs. And then if you plot that against lifespan on the bottom, so the normal lifespan of a species, you see this very clear relationship that longer-lived species have lower rates of mutation accumulation, consistent with what we were just seeing, that when you have mutations that lead to more mutations, you end up with shorter lifespan. So this is, in some ways, really strong evidence for a role of DNA repair in aging. But it's really important to remember here that DNA maintenance being necessary for aging at the rate that we are used to is not the same thing as DNA repair being the limiting factor for aging, and that sort of increasing the amount of DNA repair would automatically lead to longer healthy lifespans. Based on everything that I've covered now, people have, of course, tried to boost DNA repair. You can overexpress different enzymes involved in the process. And so far, that has not worked. People have not made, a longer-lived mouse by increasing the amount by overexpressing Cockayne syndrome A or B, which are the 2 proteins that are mutated in the patients. So why is that? There's a lot of different potential reasons. You could say, oh, well, DNA repair is a coordinated effort of a lot of different enzymes, and just one overexpression doesn't necessarily give you boosted function. But it could also be that there are multiple factors that are sort of limiting our lifespan, and that functional DNA repair is one of them, but not the only one. And therefore, fixing it does not help us until we also fix a bunch of other things. I want to go through a few specific sort of instances of DNA. I don't think I can give this talk without talking about telomeres. Telomeres were discovered again in the last century, and they're these ends of your chromosomes that have these repetitive regions, and there is a lot of interest, or there was at least a lot of interest from journalists in sort of saying, oh, this is the clock. Like we have these things and they get shorter every time a cell divides. So apologies to journalists in the room. I'm sure you haven't written this. They get shorter. And so this is like the clock. This is how aging works. And this was in part based on, research from Leonard Hayflakes, who found that, there's a limit to how many times a cell in culture can divide. And that limit is in fact mediated by shortening of the telomeres. They get sort of senescent. The problem with that sort of jump from fibroblast in culture to our body is that the different types of cells in our body, some of them divide a lot and some of them don't divide at all. And so if indeed telomere shortening was like the thing that was holding us back from like indefinite life, then you would not expect brain aging to be a thing because neurons are not dividing, right? You would expect that all humans would die from either like skin or intestinal diseases, because those are the ones that are constantly dividing, right? So telomeres as a clock, and like this is the thing, the single cause, is not real. And we should just stop writing these papers or articles or whatever. However, that does not mean that telomeres don't have a role in aging. And so there's a bunch of research on what happens when you have short telomeres. So this is sort of the structure of a telomere, where it's the end of the DNA is rolled up on itself. And the reason this is the case is because your body is keen on sensing, oh, there's a break in our DNA. And so since your chromosome is sort of a one-dimensional line, the ends look a lot like breaks. And so you don't want your body freaking out. So you roll it up, you package it in a bunch of proteins, and then now we're good, right? It's sort of like the end of your shoelaces. But that can fail. When that fails, you get exposure to this like loose end and your body starts freaking out, or your cell starts freaking out. And so there's a lot of things that happen when you have short telomeres. You get DNA damage responses. Sometimes cells go senescent. And all of this can lead to sort of, again, a dysfunctional cell that is not helping the organ that it lives in. There is also a correlation between shorter telomeres and shorter lifespan, right? So in general, if you do tests of blood or other organs, if telomeres are shorter, there's a correlation, but not perfect predictiveness at all with shorter lifespan. So it does seem like there is a role here. It's just not like the simple aging clock. The other thing is it's really hard to measure telomere length, and most measurements of telomere length are done on blood samples because people are more keen to have their blood sampled than like a heart biopsy. And the length of your telomeres of your blood cells, which do divide heavily, is not necessarily the same as in your organs. So just sort of caveat for anyone in the audience who's having their telomere length measured. The last thing I'll talk about for DNA is that it's not just the exact sequence, the bases that are in your genome that changes with age. The way that your genome is organized also changes with age. So this is sort of a cartoon of the nucleus of a cell. The line is your genome, and all the blobs are the different ways that proteins bind to your DNA in order to keep certain regions open and other regions closed. Because if you think about it, your eyeball and your skin, they have the same DNA. Why are they different? Why are these cells different? Because they're running different parts of the code. You have this massive sort of source code in your genome, but then only certain programs are running in a given cell type. And that's determined by sort of the environment and by how the different parts of DNA are exposed to transcription. So when that's all well organized, you have specialized cell types, right? With age, a lot of the systems that organize this, the way that the DNA is bound to the edge of the nucleus and the parts that are open and not open, there's lots of research showing that this basically loosens the sort of tight compartmentalization that happens in youth is to some degree lost. And going back to our sort of company analogy, right? Like if each team does not know, if they're unclear on exactly what their scope and role is, then you get a lot of people that are doing sort of a mess of a thing. And in that way, you could have cells that are misbehaving not because their DNA was mutated, but because they're reading the wrong parts of it. They're sort of like executing the wrong tasks. There's at least one disorder called Hutchinson-Gilford progeria syndrome, which also produces these accelerated aging phenotypes, and it's caused by a dysfunction in one protein called lamin that sequesters or sort of tethers DNA to the edge of the nucleus and keeps it organized. And so you have here the nucleus of a cell from a normal person and then Hutchinson-Gilford progeria patient. And you can see these blebs where the DNA is supposed to be tethered at the edge. Here you see this sort of green-red overlap, but the whole structure is misformed. And the patients obviously look like that. So that might suggest to us a broader implication of like it's not just the mutations in your genome, but it's how well is your cell able to execute sort of the specific function based on its genome that is able to produce these sort of accelerated aging situations. So yeah, in conclusion, DNA definitely matters for aging, and as I'll say ad nauseum today, it is not like a single cause explanation. It is not that all bad things arise from DNA mutations the way my sort of PhD lab would have it. So let's move on to proteins. Proteins are what do most of the things that get done in your cell. They arise from DNA getting turned into RNA, and then there's specialized protein complexes that read the sequence of the RNA and then create these long chains of amino acids, which then have to fold into an appropriate structure in order for you to have a functional protein. And so this is, you talked about AlphaFold, right? This is about, okay, here's the sequence. How is this going to fold up, right? Like what's going to be the shape of your tangle of chords, like after your travel trip? The ability to predict that is kind of crazy and cool. And the fact that this happens regularly in the body is kind of crazy because you have this just sort of mess of, at the top here, you can think of like just an unfolded strand. And what we want is that the proteins end up in the sort of energy minima, and this is all sort of physics, thermodynamic stuff, the energy minima where it is folded into the native state of the protein. You have that on the left here, highlighted in green. And that can happen, but a lot of other things can also happen. You can get this amorphous aggregate, like charging cord situation in the middle, and you can also obviously get specific forms of protein aggregation like amyloid that happens in Alzheimer's and other diseases, where the structure of the protein that has different parts that like to bind to each other ends up in a useless state, basically. And the body has a whole bunch of mechanisms that mediate this happening correctly. So these are called chaperones. They're basically proteins that bind to other proteins. There's dozens of different kinds of these. And we're producing a gazillion, that's a technical term, proteins per day, right, in each of our cells, and they're all mostly actually folding into the right shape. So that's a miracle, and sort of as complex as all life on Earth just happening within one of your cells. It does not always go well. And so the body also has mechanisms that try to clean this up. Before we go to that, I'll just mention again, back to fibrosis, this happens in the cell and it happens outside of your cell. So the proteins that are outside of your cell are also changing. But going back to, like how this affects the body, we produce this stuff and we produce also like the whole organelles in the same way. This is just one layer of organization higher. And we have this constant production, and then we have this constant turnover of the things that do not end up useful. In this image, we're just looking at mitochondria, which are specialized organelles inside of your cell that are responsible for producing ATP, which is the universal fuel, US dollar, of like cellular function. I'm sure you're familiar with, to some degree, with mitochondria. The point is you have lots and lots of mitochondria inside of your cell, and they're constantly fizzing and fusing. to create a pool of organelles. But sometimes they get messed up in various ways, and we're not going to go through all of this stuff, but that was a lot of my postdoc. And the body has a mechanism where when you have sort of mediocre mitochondria, they tend to split into a really shitty mitochondrion and a good mitochondrion. And then the really shitty one gets degraded by what's called mitophagy. And so this is mito-eating, where the cell encapsulates the shitty mitochondrion and then fuses it with what's called a lysosome that is just a tremendously hostile environment that is acidic and full of enzymes that degrade everything in your cell. And so you just break down the whole thing, feed the components back into protein synthesis and so forth, and you end up with healthier mitochondria. So this process happens for mitochondria, and it happens for different parts of your body. You have mitochondria on the top here. Lipids, so the membranes of your cells undergo this. Aggregates, so your misfolded proteins undergo this process. And the lysosomes themselves are sort of continuously turned over. And then if you look at the right side, which you can't see, but on the right side, you'll see a list of diseases. You can imagine a list of diseases that are associated with failures in this pathway. On the left side, you can see what's happening inside of a cell when you have a failure in this pathway. Because there's millions of events happening for each of these mechanisms constantly. And when they are not appropriately balanced, you end up with accumulation of something that is not as functional as you want. And in many cases is sort of taking up space and triggering signals that stress the cell out. So cells have lots of different sensing mechanisms for something is wrong. That's what we talked about back with the telomeres. Here is a loose end. That should not be a thing. Let me sound the alarm, right? And that's a good thing. Again, back to if you see some whatever, your trial balances aren't adding up or getting a lot of returns on your product, something is wrong. We need to go investigate. But if things stay wrong and are wrong for an extended period of time, that can also lead to the whole cell either becoming senescent or in some other way getting to this sort of catastrophically dysfunctional state. And so turning things over is, there is clearly an important thing going on here. There are lots of ways where if we induce these mechanisms to fail, you will get accelerated aging of different organs. But much like the DNA repair, we have not been great at making better proteostasis organisms and making things live longer by increasing the turnover. Again, like the DNA situation, this could be because we have, there are many components, and this is a thing that needs to be in balance. And, if you just start turning too much stuff over, that's also bad for the cell. And so it's just really tricky to get it right. That could be the case. It could also be the case that, yes, this is also a limiting factor for how well the cell can function, how resilient it is, and how long the organism can live, but it is not a single factor. And I think that's, if we think evolutionarily, it makes sense that we have not evolved to have just a single strategy that fails. If that fails, we die. If that was the case, if we were an organism that's just this is the way that you're going to die every time, there would be a evolutionary pressure to sort of make that single thing better, right? And so in evolution, we talk about evolutionarily stable strategies where a small change doesn't generally just take things in a completely different direction, but instead they're sort of brought back to an equilibrium. This happens in ecology, is it possible for if if a single member of this species has a mutation that makes them breed more, they would just take over the entire population, and that would be the only thing that exists? Or are there compensatory mechanisms like this multifactorial situation, but also sort of group behaviors that mean like, oh, that's not actually going to happen because then you spend more time breeding and then you get eaten by predators and something like that. The same thing is true in cells. Of course, you don't want the whole thing set up so that a single mutation leads you to become dysfunctional. There's a bunch of that in the systems medicine, and that's probably part of why we have autoimmune diseases. It's because if a mutation in a cell makes it sort of like an over-eager insulin producer, we want the immune system to wipe out that cell, otherwise the coordination of the whole system from secreted factors gets messed up. And of course, it's true in society as well. That's why we have concepts like honor and so forth, right? So that you don't have a situation where a single person can just start cheating and then get an unfair advantage or whatever. There's no fair, but an advantage from that to the detriment of the group. So anyway, I think all of this points us towards there's probably evolution of multiple factors to keep an organism alive. We know that can lead to great differences in lifespan. We saw that in the beginning. And the question is, what do we sort of do about there are multiple limiting factors here? We'll get back to that in the last section. Before we go there, I want to talk about metabolism. And so you saw this plot before. This is the worm that lives twice as long from the mutation of a single gene called DAF2. And this gene is, as I mentioned, in the pathway that senses insulin. And it turns out that whenever we find something that greatly increases lifespan, I would bet you at least 8-1 that that gene is somehow involved in sensing nutrients and regulating the body's response to nutrients. To go through a few more examples, there's a drug called rapamycin, which you may have heard about, that I would say is the strongest, most robust sort of medical intervention that we have for lab animals.
How many folks here are taking rapamycin? Raise your hand.
Okay, great. So, so rapamycin, for those who aren't, the other 70 or 80%, it is an FDA-approved immunosuppressant. So we use it for organ transplantation because when you take a high dose of this, your immune system gets dramatically weakened. This does not extend your lifespan. But the stuff that's used in the aging field is a much lower dose, which here's data from mice. And this is an interesting study because they were not fed rapamycin until they were 600 days old, which is sort of middle age for these mice, right? So this is a late stage intervention. It's not one of the mutations where even from birth, your body is running in a different way, right? And so this is more applicable for human lifespan. And you still had a significant increase in the final lifespan and the health of these mice. And this has been replicated a bunch of times. There's early data from Adam Salmon in primates that was, I've seen it presented at conferences. I don't think it's published yet. But yeah, rapamycin is really robust. Consequently, there are some clinical trials for the use of rapamycin for purposes of aging. I've listed the 7 that I am familiar with. Most of these are actually sort of RCTs, like controlled clinical trials with a control group. At least one of them is sort of observational. And 3 of them were funded by our nonprofit, which is just to say, like, there is opportunity here. The dog study, Triad, which is in a large population of dogs, and the idea is, well, obviously for you, the idea is maybe we can give rapamycin to dogs and they will live longer. The people who started this are all dog owners. The other idea is that dogs don't live as long as humans, and so we might be able to get a strong signal there that suggests, yes, this will actually work in humans. The rest of the trials are in humans. They're focused on different, some of them are in cognitive function. One is in periodontal health on the basis of sort of mouse results there, where their oral health was improved a lot. One of them is in female reproduction, measuring anti-Mullerian hormone as a biomarker. But the idea is that there are a lot of different things that improve in animals. We have a lot of different studies. This thing is the most ready for prime time, I would say, out of any drug from the aging industry. But you need the right dosing. And so I think that's why Impetus funded multiple of these trials, right? We want to find out what is actually safe and effective dosing in humans.
So if you're interested in that, you can chat with Dr. Messier or George from Fountain and see the protocol that we take. This can be prescribed for you. And again, the data is going to be coming, but I consider this a rather reasonably low risk, high potential reward, and we're constantly making those trades in our lives. So,
right. That was like, I forget who mentioned this, but I think you mentioned this. Like the FDA's role is to have nothing go wrong, but adverse events by inaction are not part of the calculus. But of course, for your personal health, they are or should be. So rapamycin, what does rapamycin actually do? It inhibits a complex called mTOR, which is mammalian target of rapamycin. And this is the insulin is the sensor of carbohydrates, and mTOR is a sort of integrated sensor of amino acids. So how much amino acids are available to the cell. When you have more, mTOR tells the level of mTOR activity tells the cell through a lot of different processes that we have abundance. Let's go, let's grow, right? I'll get back to this one in a second, but just this framework that I'm talking about. When the cell has scarcity, it wants to survive. Like if we think of, let's oversimplify life a little bit, right? The point is just to make as many babies as possible. That's not my own purpose in life, but from evolution's perspective, just spread your genes. That's the idea. And so if we think about like how do you do that most effectively, assuming that you don't have a perfectly safe and constant external environment, well, sometimes you don't have safety and nutrients. And when that happens, your best strategy for making babies is not to make babies. It is to stay alive until you have an environment that's conducive to reproduction. And so that's sort of an evolutionary hypothesis, I should say, on why are there these things that inhibit nutrient sensing, tell us that we have low nutrients, that make us, at least us as in model organisms, live longer. It's because stay alive now and then reproduce later. And so there's a bunch of different functions in the body, including the cleanup, sort of protein cleanup and organelle cleanup stuff that I talked about earlier, and also DNA repair that gets upregulated when you have conditions of scarcity. This was just caloric restriction, which I'm sure you are also very familiar with. This is the oldest intervention from the aging field. It started with Reindrup in like the 50s. There were even early human studies in the biosphere in Arizona. And the idea is that, yes, limit nutrients, you get protective responses. These are some rhesus monkeys. The one, well, you can guess which one. They're both 28 years old. One of them has had their diet restricted to 30% less calories than the other one, right? And on the right, you're seeing the not the survival, but the age of disease-free survival in two different studies. One was at the NIH and one was at the University of Wisconsin for cohorts of rhesus monkeys. So in both studies, you have an improvement for the primates that are on this calorically restricted state. And this has been sort of replicated in mice and flies and all over the place. So caloric restriction, it's the big thing. There are a lot of people looking at what is the gene that causes this and so forth. And some of that overlaps with, and some doesn't, drugs like rapamycin. So, okay, should we be calorically restricting ourselves? As Peter mentioned, I spent 4 years eating every other day and ended up writing a book about that because Danish journalists found that interesting. Why don't I do that anymore? And should we be doing that? At the time, this was during my PhD, I was like, well, look, I'm reading about all this stuff. If I'm serious here, I should be practicing this, right? Like, why am I not doing that? And so I did that. Maybe I ate slower. It's hard to tell when you're 24. But the reason why it's tricky is that if we go to intermittent fasting, which is what I was doing and which is sort of similar to caloric restriction, this is a really old paper, as you can tell by the way that the hand-drawn data looks. But it's testing different rates of fasting in rats. And so each box is sort of the lifespan of one of their rats. And on the top line here, you have the controls, and then the top line, they're fasting 1 day in 4, then 1 day in 3, then 1 day in 2. And you have sort of a spread, there's a trend, right, where like the ones that are fasting more often are better off, but it's kind of hard to tell. And so the question here is, what is the right amount of fasting in a human? Is it 5-day water fast? Or I forget, where did you go? Here you go, right? Is it a longer? In rodents, it seems like every other day or maybe 1 day in 3 is good, but like rodents have a way different metabolism than humans. So like, what is the right amount in humans? We don't really know. For caloric restriction, so this is just set reduction in calories. Here's 2 different studies in mice and in flies. And so normally, most of the research that's done in the lab is on like black 6 mice. They're genetically identical. So these studies were not. These were done on genetically diverse groups of mice or flies. And then they put them all on the same amount of caloric restriction. And on the left, you have color-coded males and females, males in red, males in blue, females in red. What you see is that for the same amount of caloric restriction, some of the genetic strains live much longer, and some of them live much shorter. It's basically a bell curve. For the flies, it's a little better. A lot of them live longer. But the point here is that there probably is, like you probably should be calorically restricted. But given that you're not a black 6 mouse, how much should you be calorically restricted is hard to say. And so part of the reason I eventually stopped doing intermittent fasting, one is sort of startups are stressful, less mental overhead. But another one is I'm not sure if I'm doing this right at all, right? Like if I'm obese, that's definitely bad, right? But if I am eating healthy and not eating a lot and exercising, it's unclear how much I should be doing this. I think if we have ways to measure our health, and if we're experiencing sort of like clear benefits, my parents are still doing intermittent fasting. They are, their cholesterol.
Can I get a microphone for Daniel here? I want to just take a second to hit this.
Their cholesterol and BMI are a bit higher than mine, and they're like getting very obvious benefits from it. So if that's the case, yeah, you should.
I want to make one statement and ask your input. I just want to nail some of these things down along the way here because this is important, right? The work done in rhesus monkeys, they reduced the amount of monkey chow by 30%. And there was a definite physiological improvement in how they look and ability to fight off disease and so forth. If you're overweight, I'm super clear. And if you have high visceral fat levels, super clear that fasting is going to provide value to you. But you're not overweight and don't have a high, what's your BMI and your visceral fat? And why are you doing this?
My visceral fat in my liver is about it's under 1%. That's
under
good.
1%.
Under 1%.
That's pretty stellar.
It's 4 standard deviations under the bottom quartile.
Yeah.
So that's pretty good. Probably thanks to the water fasting. So one thing I'd say is caloric restriction and water fasting are 2 very different things. Caloric restriction, if you reduce something every day by 30%, you're increasing your insulin sensitivity. That's good. You're reducing your insulin spikes. That's good. That kind of stuff, right? Water fasting, prolonged water fasting, why I do that is because it increases your autophagy quite a bit. We're talking multiples, right? That's not what caloric restriction does.
And autophagy is?
And autophagy is essentially self-recycling, self-eating of the cells and recycling materials, right? And it appears that autophagy selectively goes after senescent cells. That is so before we have senolytics, autophagy is the way to go. That's why I do water fasting. And we don't have any way today to kind of measure how good or bad it is for you. So I'm just kind of going with empirical, right? It's
And
kind of,
Helen, I'm going to ask you for any input on or advice on this here.
so I go with empirical. So what I wanted to say, I've made a bunch of my friends at this point do 5 to 10 days water fasting every now and then, and I'm just kind of collecting the feedback, and almost everybody always says, oh my God, I feel so much better, so much younger. I do feel 10 years younger, blah, blah, blah. There was a scar, on my skin that's now gone, things like that. That's what people actually report, things that you wouldn't even expect. And people actually do look better too.
Does
And
anybody
I'm
else
fit,
here
so-
do extended water fast? Okay, Umang, any comments from you? Thank you, Daniel.
Yeah, for me, it really works. So I started off with 1 day a week, every week. And then that was every Monday. And then started Monday and Tuesday. And now I do Monday, Tuesdays, Wednesdays.
Oh, every week?
Every week.
Water fast?
Yeah.
So what's today? Today's Wednesday. You're not
Yeah.
eating today?
No, I am. Today
Okay,
I
you're
am.
not
Because I'm
traveling,
traveling.
yeah.
When I'm traveling,
Yeah.
I make the exception.
Yeah.
But when I'm at home, 2 to 3 days a week, it just completely makes me feel very high on energy, helps control weight, and makes me actually feel and look better.
By the way, one of the things, if you do intermittent fasting, you start to like vibrate on energy because your blood is not being deviated to your digestive system, right? Helen, how do you think
It's the
about
ketones as a source of
and
energy.
ketones.
So you're running on ketones, which is better than glucose for brain.
Yeah.
And just for the record, Peter is denying you lunch, not me, when we go over at the end. But that's supported, right? That's what we're talking about.
Yes, yes. By the way, we're going to cut back on the budget for food this week. Helen, you're a functional medicine doctor, and functional medicine is looking at root cause operations. How does the functional medicine field think about this?
Yeah, I think it's from a functional medicine perspective, it's all over. I have my opinions on it. And I think we're not looking enough at the timing of when we get the calories in. So we are subject to circadian rhythms. We have clock genes in every cell of our body, and those clock genes regulate everything, including our metabolism. So they regulate our sleep-wake cycle, wake us up, put us to sleep, but they actually regulate our metabolism. So in the morning, we're actually a lot more insulin sensitive than we are in the evening. So if you eat the same calories in the morning, it'll be processed by your body much more efficiently. You're not going to produce as much insulin when you take in those calories versus when you eat those calories at night, you're going to have much more calorie resistance. So I think a lot of the studies that have been done on the calorie restriction in animals, for example, if you calorie restrict mice and then you give them that food, they're going to eat it right away. So the timing of the food depends on when you give it to them, right? So, but if you space it out, it matters. So looking at those animal studies, we have to take into account when they ate the food as much as how much food they ate. And so I think it's a really important thing. So the way a lot of us do intermittent fasting, you do 8 hours of feeding or 6 hours of eating, we tend to skip breakfast and lunch oftentimes and just eat dinner. And I think that's a problem because we're eating at a time when our body is much more metabolically unhealthy, so to speak, than in the morning. So I also think it's important to stop eating a number of hours before you go to bed. That's critical. That will stimulate autophagy because we do most of our cleanup at night when we sleep. So you don't want our body processing calories at night when you sleep. That's when you want to fast before you go to bed. So I think, there are definitely a lot of studies on humans, the metabolic, especially if you're overweight or obese, like you said, there's a lot of benefits to really reducing calories and doing the intermittent fasting. But I think you really need to look at timing.
Vibhab, let's take one more question and we'll continue on.
So actually, I did exactly that. So I read circadian code. I know we're meeting Dr. Panda in San Diego. So I've been following that for a while, and I just found it's the only change I made. And I'm not even doing like a 16-hour kind of thing. It's just a simple 12 to 14 hours. And I lost 10% of my body weight just with that one
you
change.
did that by fasting between when and when?
It's just like having a relatively early dinner, a relatively late breakfast. I'm having enough meals a day. Like it's the only change I made.
When do you have dinner?
By like 7:30, and breakfast maybe by like 9:30. just with that one simple change over the course of a few months, like I've lost 10% of my body weight, and it's maintained that.
Which 10%? Just kidding. Okay.
I mean, it's also interesting, right? When we do restrict the time of when we eat, we also tend to eat less calories, right? So the two definitely go hand in hand.
I started this 15 minutes early to give myself a little bit of margin here.
Oh,
so.
okay.
Anyway, thank you.
Cool. So yeah, I think the point, the main point, like this is all great conversation, and I think the opportunity to tailor that specifically to you, that's like kind of the takeaway here, right? Like there probably is a correct amount and timing of eating, but it is hard to say from the animal studies exactly what that is. Like we don't have the data yet, right? And there are some human studies that are happening on intermittent fasting and so forth, but not very many. And there's a lot of stuff to measure, and there's a lot of variables, right? You talked about like circadian rhythms, right? Well, mice are active at night. So what should we conclude from like this time is the best time for a mouse to fast compared to a human?
Can we reset the clock so it ends at 12:30 instead of it?
Cool.
Okay, great.
Nevertheless, I think it is indisputable that when, like in the aging field, a lot of the things that we do come back to nutrient sensing and activating genetic pathways or inhibiting genetic pathways that relate to nutrient sensing. Like if we have a success story so far, this is definitely it. And the good news is there's at least something that's applicable here. And there are medicines that are sort of being created on that basis. In terms of what we have not yet succeeded at, but many people in this room hope to succeed at, I want to talk about, okay, so we went through a lot of different biology, and we kept saying, well, there's multiple things going wrong, and we may need to do multiple things at once and so forth. That's complicated, especially in terms of sort of regulation and drug development. It would be nice if there was a silver bullet. And so far, I've sort of said there's not a silver bullet. And I think as far as we know right now, that seems true. But let's talk about the shiniest looking bullet in the pile for now. And so, as we mentioned in the beginning, you can make a baby and the baby is young. So there's something there where biology is able to take a cell, turn it into an embryonic stem cell, turn it into a whole organism that is young. And we've, 2012 Nobel Prize, we've been able to turn older cells, like a skin cell from your body. This is what I think your nonprofit is partly funding at the Mayo Clinic. We can turn cells into stem cells, and we can turn those stem cells into functional cells. And in many cases, they have sort of like a younger phenotype. And so we've done that to cells in culture using these Yamanaka factors and other methods. We've also been able to do that in animals. So we can take the animal and induce reprogramming in those cells. Now, the relevance for the aging field really ramps up in 2016, where a paper came out from San Diego. Maybe you'll meet Juan Carlos. But a paper came out that said, okay, let's just do like a little bit of reprogramming. Right? Like, let's just do it on the weekends. That was roughly their protocol, right? 2 days a week, we're going to trigger these reprogramming factors. And so we're not going to recreate the ending of Akira, if anyone knows this anime, where the entire mouse just turns into an amorphous blob because everything is now a stem cell and we've completely abolished all these specialized functions that the cells were trained to do. That is not beneficial for lifespan. But we're going to do it slightly, and then we're going to see if that benefits the animal. And so they reported one of these progeria accelerated aging DNA damage mouse strains. They reported an increased lifespan from this regime of partial reprogramming. And they also reported sort of benefits in other tissues for wild-type mice. They didn't have lifespan data for wild-type mice. So this is currently a hot topic in the aging field because it may be a silver bullet. It may be we see all of these different aspects of aging that all get rejuvenated when you create an iPS cell, this induced pluripotent stem cell. And so if we could do that, maybe we can take all that complexity and just like wipe it all away, right? What is the current state? Can we do that right now? There was this 2016 paper. They had they have mutant mice. They did not have any wild type mice. Juan Carlos said they were doing wild type mice, but there is no paper yet on the wild type mice. In 2021, David Sinclair's lab published a paper in Nature where they focused on sort of regeneration of optical nerves. If you go into the supplemental data, you will see that they also they had their AAV carrying the OSK factors. They did administer that to like the mouse at large, like the whole organism. And they did measure lifespan of those organisms because they classify the age of death at different times, but there is no lifespan curve. If you read between the lines, this means there was no lifespan extension. So there were some effects in the eye, but they were not able to produce sort of like actual longer-lived mice. Then there's a paper from, there are a lot of people involved in this. Some of them were at Genentech. I forget all the affiliations. They did look at lifespan data from wild-type mice that had received sort of a partial reprogramming thing. They saw maybe some younger skin using sort of molecular sequencing and some phenotypes, but the lifespan did not improve in the duration of this study. They didn't have a ton of mice. There is a paper actually from Rejuvenate Bio, so you can grill Daniel a lot on this paper, where it's a published paper now, where they did say, okay, we have these mice and we're showing a lifespan effect using AAV-delivered Yamanaka factors. So yeah, find more detail because the paper didn't have a lot of detail, and this was quite recent. And then there are some other papers that have been published that say, oh no, actually you do end up with a cura, like you get a lot of liver cancer or something. So
Martin, I think we need to take 1 second because we haven't yet to talk about, again, the Yamanaka factors, and when you're talking about delivering these by AAV, can you just expand on that one moment?
Yeah, sure. So what are the Yamanaka factors? They're named for Shinya Yamanaka, who got the Nobel Prize for this. They are 4 proteins, 4 transcription factors. So OSKM, M is MYC, and K is KLA4, and OCT, and so forth. So they're transcription factors that are active during development normally. And so these 4 proteins. And so these papers, the Masip paper and the Liu paper, they delivered these in the form of a gene therapy. So an AAV gene therapy you'll see, it's basically a virus that normally carries a piece of DNA cargo that makes more virus. But back in, I think even before the 90s, Molsky repurposed this virus, basically like, oh, I ripped out all the sequence and I made it carry my proteins of interest. And so this is the basis for these miraculous gene therapies that have restored basically vision, hearing, name your miracle, cured SMA kids who would otherwise die within a couple of years, and they seem fine. So this is an amazing new drug modality that has been used in humans, not to reprogram stuff yet. But so you're overexpressing these proteins, and that's what's causing the reprogramming. And then they have different mechanisms for controlling, when is it active and when is it not active. So, go ahead.
We'll go into more detail on this as we hear speakers, but this is one of the areas of epigenetic reprogramming. We have 3 companies you'll be hearing about, all 3 backed by a group of billionaires, with can we reverse the aging of your epigenome, which genes are on and which genes are off, right? So we'll go into more detail about this. But so far, the belief is that this is not the magic bullet.
I would say it is not working. It has not been reported to work yet in the way that we want, which is a magic bullet, right? Which is like you're just younger in at least a broad swath of ways, right? So there is a lot of sort of interest here because of the potential. And I would say the potential has not yet been realized, but the yet is very important because we don't have any evidence that like this cannot work, right? It's easy to confuse, like this is bullshit, like this thing is not really like this is not a good paper or it doesn't actually work and all this kind of stuff. I think it is fairly clear that like people haven't made really long-lived mice using this method, or if they have, they've done it quite recently and not told anyone. But that does not mean that it cannot work. if you go back to the original Yamanaka factor paper, how did he find those 4 proteins? Well, the answer is they brute forced an absolute ton of different combinations of proteins to see if anything could create a stem cell, right? And so there's a specific combination of things in a specific environment that works for this purpose. Now we're trying to do a different thing. Now we're trying not to create a stem cell, but to rejuvenate other cells. What is the right combination? And so that's where the companies that Peter mentioned, and I think you'll hear from actually all of them today, so that's convenient. Altos later, super well funded, kind of a behemoth. Retro and New Limit, sort of only regularly, lots of funding, human level, lots of funding. And Joe's over here, who's the CEO of Retro. So you'll hear from all of these, right? So there's a lot of funded stuff, and there are people who are exploring, okay, what is the right combination of things and so forth, right? So I would say this is definitely an area to keep a close eye on. There may be a silver bullet. We haven't found it yet. And Joe can disagree on his time if he wants, but I don't think he will.
I think the answer is yet still.
Right.
I think there's something very important to make a statement at this point. The field of age-related research, of regenerative medicine, whatever name you want to give it, is a relatively newly funded field. You heard the numbers before from the 400 million for government funding. You've since then had Evolution out of Saudi with $1 billion a year funding. We've launched this $100+ million X Prize, and we've seen extraordinary people like Sam Altman and Brian Armstrong and Yuri Milner and Jeff Bezos and a multitude of individuals begin investing in this field. So there's far more capital. And the other thing I would point out, and Martin, I'd love your view on this, is that traditional government funding is risk averse.
Yeah, for sure.
And peer review means that your peers don't want you to disrupt the current model.
Yeah, and not everyone who does research has, and in fact the minority, have a very specific sort of like, we are going to have this impact on humans in 30 years, the way Norn's mission statement is, and a lot of people here are interested in, right? But like, that's not the mission statement of an academic institute that does research on this, right? It relies on the idealistic motivation of individual researchers to actually really try to push treatments that work, as opposed to studying the basic biology, which is a absolutely critical function for, like, all this came out of just like, I don't know, can we do this? Curious, right? Like, if we didn't do that, we wouldn't have any medicines. And so we should absolutely not stop having curiosity-driven basic research, but that's a different thing than, we need a solution that works in humans, which is incidentally why I left academia. And I do think that more funding for that type of work, that's exactly why we started Impetus Grants, and you'll hear from Lara, I think, tomorrow or later, who's the program director of Impetus Grants, right? It's like, let's fund direct money specifically at the things that has the potential to really impact us in sort of the timeline of decades. All right, so let's move on from cells to coordination. This part is going to be a lighter touch. You may end up remembering less from this part, and that's largely because it is much more complicated, as we arrived at in the beginning, right? When you talk about a lot of different things that can interact in different ways, it is hard to find the answers. I think if that's the only thing you take away from the next session, great, because that's one of the most important things in the aging field. This is, sometimes I jokingly call this the greatest lie in biology. This is sort of the apoptosis cell death pathway from a classic textbook. The pathway is not a lie. Apoptosis is actually a real thing. What is a lie is that you have this deterministic set of arrows that make things happen in an organized way. Here's a slightly less oversimplified apoptosis pathway. Same biology, same thing. It has more of the relevant factors, but probably still only 10% of them. And every arrow is some sort of feedback mechanism, right? And you can see some of them go in circles, and it's pretty complicated. It's important to remember that this is how biology works. Everything has multiple causes. Everything is dynamic. Everything happens within a given system. And that makes it hard to study, right? Like that's why biology, the studies take a long time and we get mixed results all over the place, apropos of sort of reprogramming. And so this is the reality that we're operating in. This means that cells, in order to do anything, they have to coordinate a lot of interacting parts. And I'll just sort of mention this as an example. In mitochondria, on the left, you have this electron transport chain. And so this is how the cell produces energy to use for all of its functions. And it involves these 5 subunits, complexes, that each involve multiple proteins. And they're handing off sort of different oxidized states of different atoms to each other in order to produce this ATP. But all of this stuff has to happen in sequence. Otherwise, you get nothing. And if you don't have the right number of the different subunits, so on the right, we're sort of seeing what happens when you have too little and too much of a given protein. If you have too little, it was supposed to be part of a complex, now it's missing. Now the complex can't even assemble itself structurally. If you have too much, you overstress your chaperones, you get aggregation. And so everything has to be just right and coordinated in the right way for even a cell to perform its basic function. So that's just at the level of cells. And then if you remember back to sort of the company analogy, life is organs communicating with each other. And they do that through lots of things that are secreted into your blood and that send signals, right? So we think about insulin from the pancreas, right? And that's telling the whole body how much nutrients do we have. And when it goes awry, we could die. We could have glucose poisoning, right? Or we could have chronic super low glucose and the cells could die. And so there have been a lot of studies, especially recently, that we've gotten better proteomics that have looked at like what changes in the blood with age. And you find a bunch of different proteins that change. There's pretty good consistency between different studies, but there's still lots of new stuff being found every time there's a new study. This is from Tony Wiscore's lab at Stanford, who's doing a lot of this. And it's not all sort of simple up and down. If you look at this curve, yellow is up, blue is down, and it's just tracking each column is a different protein. So some of them go up, then down, and then some of them just go up, and some of them go down. There's just like a lot of changes happening in the blood. And in some cases, those blood changes track with diseases. And so here they're looking at some of the changes that change with blood with age, and they see how well does this correlate with sort of self-rated health or incidence of individual diseases. And we'll get back to the idea of sort of aging clocks in a little bit. But the main point here is that your body is a coordinated system of different parts that are working together. And the signals that go between, there are a ton of changed signals in the blood, which is coordinating your organs. And so there's definitely something to find here in terms of like what is good and what is bad, and what if we just injected this protein and so forth. You're probably aware of the concept of heterochronic parabiosis. So this is heterochronic, meaning different ages, parabiosis, sharing of blood. And so these are experiments, many of them from Stanford and also Harvard and other places, where, okay, you stitch 2 animals together, or later on, we just inject serum from one animal into another, and then you've seen sort of benefits on a range of different tissues. The idea here, of course, is that there's maybe something good in the blood, or maybe something bad in the old blood, or maybe both. And if we can find out what it is, then we could just sort of like recreate a youthful circulating environment, and this might have benefits to your whole body. I think the short version here is multiple companies have been started to find out what it is. So far, none of them have like produced a treatment that sort of has shown to be effective. I know that Retro is doing something here, and I imagine Joe might talk about it. They're actually trying to remove bad stuff from circulation. There's definitely something here. And the question is, okay, you have these 5,000 things, and they're not independent variables. Like more of one thing might be good only if another thing is high or low. How do you figure that all out? And we'll get back to the concept of like aging needs more tools to figure all those things out in order to make rapid progress. One thing that we can talk about in a bit more detail is sort of the immune system and inflammation, right? So obviously with no immune system, you're toast. But even outside of that extreme, the immune system is important and declines with age. And so as you age, sort of moving to the right here, there are multiple things that happen. On the left, you sort of see this myeloid skewing. So you have different types of immune cells, a lot of different types of immune cells. And basically the way that they get produced and the ones that you end up with when you're young is different than what you end up with when you are old. And so you get this sort of imbalance that leads to altered function. You get a range of different sort of responses of the immune system that get attenuated with age for reasons that would take me another hour at least to talk about. But that's why, for example, when you get a vaccine when you're older, it's not necessarily as effective. And you also are much more liable to have and to die from infectious diseases when you're older. And then finally, as I mentioned earlier, there are cancer cells, there are senescent cells in your body that the immune system is supposed to recognize and remove. And when that doesn't happen, they accumulate more. And insofar as inflammation, chronic inflammation, is actually one of the things that's sort of dampening your immune system, that creates sort of a self-fueling loop, which you see on the right here, where senescent cells are secreting cytokines that exhaust T cells in your immune system, and then that leads to lower immune function and so forth. So yeah, everyone knows we have sort of reduced immune function with age, but also the immune system probably limits the rate at which we age, and therefore sort of as it declines, you get other things that go wrong as well. I mentioned these circulating inflammatory factors. Inflammation is, a lot of things. There's a term called inflammaging, which is sort of increased inflammation with age. These are some things that you might have measured in your blood or probably will measure in your blood at some point in this week. There are different cytokines that are basically sort of triggering the immune system to be more active. And we see that with age, there's sort of a gradual increase in the circulating levels of these. So this is just what's in your blood at all times. And that is not good. So for various cytokines here, there's a lot of associations between sort of elevated levels of these and activity and age-related diseases. So in general, what we want from the immune system is sort of like targeted missiles, right? That go in and blow something up that's bad, like a cancer cell, but not something that's just like indiscriminate, rapid fire shooting all over the place. And we lose that with age because you have this chronic inflammation. I'll talk about three main sources of this chronic inflammation, but of course, this is oversimplified. There's a lot of reasons why you get inflammation. We already talked about senescent cells, and the accumulation of these cells is one source of sort of elevated cytokines. And then we also talked a bit about adipose tissue, which is another source. So in the middle, your fat, and especially your sort of the fat around your organs, your visceral fat, as your lipid reserves go up and as you accumulate more fat, the local environment that the adipose tissue sort of requires to just sustain the load of like, hey, there's a lot of weight here, requires it to remodel and like create this fibrosis around itself actually just to have sort of a scaffolding around the fat cells. And the signals that it uses to make that happen are a lot of sort of similar to senescent cells. They're these things that are inflammatory and can create fibrosis. And so when you have a lot of fat for a long time, basically your whole body is receiving a like, hey, let's make some more fibrosis signal. And so being overweight is associated with a lot of these diseases for that reason. And then the last part, which is kind of understudied, I think, in the aging field, is infections. And not just like, oh, I got COVID now or the flu or whatever, but you also have viruses that are sort of sitting dormant in your cells, like cytomegalovirus. And you might have sort of chronic exposure to bacteria. One area that's been sort of increasingly of interest is the connection between your gums and Alzheimer's in your brain, and where there's this pretty clear correlation between having gingivitis, so like receded gums, and Alzheimer's. What's going on there? And it's not fully fleshed out yet, but on the right, you're just seeing the increased bacterial load in the mouths of people with Alzheimer's, and a tau load, which is one of the proteins in Alzheimer's, that sort of correlates with the amount of bacteria present. And so there's definitely something to be explored more here. This idea that we're exposed to these chronic infections, and that's accelerating the sort of degenerative and inflammatory environments that exist in the body.
I want to just echo this. So for me, I am heads down on every aspect. And again, through Fountain, I've just gone through a whole set of testing on my oral microbiome and what bacteria are there. And, I found a number of particular strains that I'm not happy to have. So I've started going through ozone treatments to try and kill those, and of course of a particular antibiotics, which I hate taking. But the downside of having that in proximity to my brain is enough that I'm going to not fool around with that. There was another point that you were making about this. It's the notion that idea of immunoexhaustion. Right, which is that our immune system, as we grow older, it's having to deal with more and more viral loads of CMV or herpes or a whole slew of things. And if your immune system is being constantly distracted by everything else out there, it's not there when you need it against cancer or against other infectious diseases. So another critical area.
Yeah. And one that I think you're going to hear more about, and that definitely needs to be studied more. The immune system is tremendously complicated. Cool. So with that, I'm going to go to the last section. We're near the end, where we're going to talk about just okay, this was a lot of biology. The motivation for us to learn what to do here is great. What's actually in our way? What do we need? What does the aging field need the most? As was already mentioned, the longevity ecosystem is growing. This is just number of companies. I didn't have data after 18, but point is, graph go up. If you plot funding, it's the same thing. So far, we've made worms live 10 times longer. That's pretty impressive. We've made flies live sort of almost 3 times or 3 or 4 times longer. We've made mice live 30% longer with rapamycin, and we have mutants that live 70% longer. And we don't have any human trials showing humans that live much longer yet. The key point here, again, is yet. But the malleability that we talked about in the first section is evident. And then, the sober reality is we don't know if we have the cure. We don't know it for fact yet. The challenges that I want to go over, one is the complexity of the system. And then the other one is the rate at which we can do experiments. And then finally, how do we turn this into a company and create momentum behind this? And I think we'll go 5 minutes over, sorry. Complex systems. We looked at this earlier. This was just one process that's happening within the cell. The reality is that we don't have these linear cause and effect diagrams. We have these complicated, we don't have what's on the left, let's do more of X, then we get Y result. That's not how biology works, unfortunately, or we'd be a lot further along. It's more like on the right, you have a bunch of different factors and you're trying to do causal interventions into that system. How do you do that? And specifically, how do you know what is the right thing to do? Few examples. We talked about sort of IGF-1 as one of the insulin sort of pathways. You can make mice live longer if they don't have IGF-1 from birth. At the same time, IGF-1 in the bone marrow goes down with age and leads to this immunoexhaustion. And if you supplement it midlife, it gets better. Okay, so is this thing good or bad? The answer is neither. It's good or bad depending on context. Same thing in the immune system. You have on the left sort of this simple idea of inflammation is bad and this thing is good and this thing is bad. And then on the right, you have just what happens if you get exposed to a bacterium. You have all these different cytokines and immune cells that get activated and then they get deactivated. And I describe it as a symphony because do we need more tuba here? More tuba is not, more cowbell, I should have said cowbell, right? Just adding cowbell is not a universal answer to stuff. You need the things working in coordination. So how do we achieve that? How do we get good answers in complex systems? And I'm not going to solve that. You can listen to Francisco afterwards. what we do at Gordian is we found ways to put many different interventions in. What we do, and also what some other people do, which you'll hear about later. But we've found ways to, if you can put in a lot of different perturbations, a lot of different potential therapies into a complex system, and then you have a way of measuring a lot of things, like how does the system respond in sufficient detail, then you can go through this loop where you do experimentation and you're learning how the system works and you're generating data sets that are large enough to actually sort of create machine learning models and so forth and make predictions around what will happen. That is a large answer to the problem. I think I'll boil this down to the aging field needs more avoidance. Needs more people who are coming up with new ways of doing things, new ways of perturbing a lot of stuff, and new ways of measuring a lot of stuff at once. If we continue this idea of let's go do a whole research paper for 5 years on this one gene and how it relates to this one other gene, it will take us infinity years to map out the whole thing. So that's one of our challenges, and that's a general challenge for biology, but it's especially true for complex biology like aging. And yeah, that'll be covered a lot by different companies today. The second thing is, okay, are you older or are you not older? How do we know the future? How do we know whether what we did was correct at the level of aging, right? And we talked about that with the fasting. We can see it, but fundamentally, if it takes us a long time to measure, this graph, right, takes us 60 or 70 years to create. If that's the rate of measurement, if that's how long it takes us to measure whether a thing worked or not, then our iteration time, if we don't know what to do and if we need to try a lot of stuff and play Edison in order to find the right thing, it's not good news, right? The rate of progress is inversely proportional to the rate at which we can measure whether a thing worked. And so both for clinical trials, but also just for the basic experiments, if we had a really good biomarker, if we had something we could measure and that tells you the future, then we could make much more rapid progress. And so a good biomarker is something that predicts future outcomes. Measure something now and we can predict the future. It has to be sensitive and specific. So it has to actually respond when we do the right thing, but it should only respond when we do the right thing. Otherwise, it's going to give us false signals. It should be something that doesn't kill us. Right? So most of our biomarkers in animals so far are not that. Ideally, it's something that we can actually interpret, so we can try to develop drugs based on what it's doing. And then it should also ideally work in both animals so that we can do experiments and find therapies, but also work in humans. So people want to make these biomarkers, and there's a lot of different efforts to make these biomarkers. I'm going to talk about epigenetic clocks because you're going to hear about epigenetic clocks. There are other approaches as well. Epigenetic clocks are sort of methylation studies. Let's take it back a bit. You have your DNA, and then there are different chemical modifications that happen to your DNA called methylation. And they're part of this, what gets turned on and what doesn't get turned on. And more than 10 years ago, Steve Horvath and some other people said, well, I wonder if there's methylation patterns that correlate with your age. And so they made these clocks, epigenetic clocks, because the answer was yes, there are methylation patterns that correlate with your age. And they created these sort of predictors that's if I just know these 200 sites, their methylation, then I can tell you how old you are pretty well. Not as well as your driver's license, but pretty well. That's cool, but again, your driver's license can do that. So the second generation of clocks were, well, what I actually want to predict is not how old you are, but whether you are or will be sick. And so new clocks were made, like PhenoAge here, that will predict sort of your incidence of getting sick. And then work continues on this at Altos and other places to make clocks that are more nuanced. So here's one from the Buck Institute where they realized that, actually a bunch of different immune cells are behaving differently with the clock, so we should deconvolute them. So there's a lot of work to sort of measure these methylation sites and then try to predict things in the future. But what do the clocks do? Generally, they're a black box. Steve Horvath just said which sites correlate with this thing. We have no idea what those sites do. A few of the clocks are trying to address that. they may be they may not be specific to aging stuff. In some cases, they may actually be measuring what is the composition of immune cells in your blood, different types, and that changes with age, but that's not all that we want to know. And so there's those kinds of artifacts. There's also how, if you went and did 5 tests on the same sample, how close are they? Well, they might be off by up to 10 years. That limits their utility, right? Like how sensitive are they? So there's a lot of work in the field on like improving those metrics. But there's something else that's even more important. And that's what we want to do with them is like if you are on the red curve here, you're going to die sooner. That's the prediction we want to make, right? We want to use these as a clinical trial readout or in our experiment. And currently the way that we test them does not test that. So the way that we test them is like, oh, here's people with HIV, they have a higher clock age, but it's cross-sectional. It's looking at a population, different populations at the same time. It's not actually making predictions. And so if we wanted to really see whether these clocks are useful to us, here's what we should do, and this hasn't happened to my knowledge in the last 10 years, but I hope Altos is doing it. Take a bunch of mice or whatever other organism, measure their ages, do the clock thing, see what's their biological age, normalize all the groups. You have a bunch of different groups, and then give them different interventions, some of which, like rapamycin, should extend lifespan and so forth. Then measure their clocks again. Predict. Make predictions about which ones are going to live longer or not. And then do the experiment. Takes a couple of years. You could start at sort of later life, since rapamycin at least worked there. And then see how often were you right. And that lets you test the specificity of your clock. That lets you like really benchmark them. This is not, to my knowledge, something that's happening, but it should happen. And someone should team up with this is the kind of stuff we do in our nonprofit, right? Team up with the intervention testing program that tests all of these drugs and say, hey, you've already got all this blood. Let's do like a clock benchmarking testing program.
So will you be here while Altos is speaking?
I will, yeah.
All right.
I think
I invite you to ask them the question.
Yeah, I've prodded them on Twitter already, but in any case. So basically clocks, a bit similar to the reprogramming. There's something really interesting here. If we could actually use these as a biomarker for aging, that would be amazing. And we don't know if we can yet because we haven't really tested their ability to predict our future. All right, finally, how do we turn this into a company? How do we get these things on the market, right? As was already mentioned, we tend to wait for diseases to manifest, and then we treat them. And then we fund, insurance will pay and so forth. Not so much, hey, do this thing now. It will prevent you from having the disease in the future. Now, there's good reasons and bad reasons here. Bad reasons are sort of like the whole dynamics of the US healthcare system. The good reasons are the future is kind of uncertain, right? So like everyone discounts the future. I'd rather have money now than the same amount of money 10 years from now, right? And insofar as we can't predict the future, it is tricky to say, like, do this thing now and spend $1 million on this thing if you're not sure whether it'll work. So I think to the point we just made, getting better at predicting, like actually measuring, did this health thing work, is a huge sort of impact for the aging field. In the meantime, what can you do? Option one, you can just not do the FDA at all and sell unregulated sort of supplements, Elysium, lots of others. You're going to get massively exposed to this. You probably are using a bunch of these already. So this can work. Like the FDA doesn't have a magic thing that makes things effective after testing. The problem is that if you're trying to do this in a serious scientific way, you're competing with people who are not, who will just completely make stuff up and sell lies. It is difficult to do science and compete, to cost-conscious consumers with someone who will just make things up. So I think that's a challenge. And that's why most people don't go this route, because the sort of return on capital is limited. You have this sort of commodity market when you don't have this FDA-enforced scientific test of does it really work. The other thing that some people are doing is going into domestic animals. Loyal is one company here, Rejuvenate is another. You still have to sort of go through a regulatory process, but it's much easier and you get readouts faster. And then, of course, you can also just say, whatever, I'm just going to sell this to people who want it, and then you have some market. But of course, most companies are not taking this route because the market is much smaller. The other option is to go after specific diseases, and this is what most companies are doing. These are Unity, which is still alive, but launched a little while ago. Um, Peter has pessimistic estimates of their future lifespan.
High praise, still alive though.
We'll get into sort of their key OA trial. And then there's BioAge, which is also clinical stage and has obesity drugs. And then there's Retro and New Limit that are in the room. And, you can correct me if I got your indications you considered wrong. This is largely from sort of the internet and stuff. But basically, there is a list of diseases that a lot of the aging companies are going after. Immune function, we talked about to some degree already, rejuvenating immune cells and getting better sort of like cancer outcomes, neuro and fibrosis, which we also talked about, right? So there's a list of diseases that are plausible, these are age-related diseases, and these are plausible things that you could go after if your drug actually improves aging. And so a lot of us are converging on these sort of lists of diseases, and the idea is then, like, Ozempic, okay, you do this one, but actually it's going to work for a lot of diseases. So it's sort of like okay, you have the risk that if it doesn't work for the first one, you might be in trouble. But if it does work for the first one, this is going to be a way more valuable drug than someone who is developing a very targeted sort of like HER2 cancer drug or whatever it is. This is the most common path. And it makes sense. And like this is the path Gordian is on and so forth, given the current regulatory environment. There are risks. It's not an easy path. Let's explore Unity for a second. And they're the only company that's not in the room, so that's one reason. But okay, how did Unity come to be? There was this paper, Clearing Senescent Cell, Lifespan Curve, Nature. It's amazing. People get excited. Art gets excited. There's a company funded to like do this stuff. They're actually at the buck sort of physically for a while. Then, well, how do we kill senescent cells? There are some drugs identified that can do this. Unity does the whole sort of drug development thing, come up with different compounds that can do it. And then they figure out, okay, well, what disease are we actually going after? They're going back to this thing of like, we need to target a specific disease. Again, papers, research, they arrive at osteoarthritis as the first thing. There are senescent cells accumulating in OA. You heard about that earlier. Let's go treat this. They run a clinical trial, and the trial fails. They did not succeed in treating osteoarthritis. How did this happen? I don't know, obviously. But I think there's one thing to think about here, which is they ran this trial on the basis of, let's say, this Nature paper. On the right, you see the paper. You have, you can improve the osteoarthritis of 10-week-old, so very young, male mice that have surgically had OA induced. And then they run a trial in 40 to 85-year-olds that have not had surgery, and they're both sexes. And so does the model represent the patient? I think that's a huge challenge for the aging field. Finding models that have actually sort of spontaneously developed these diseases that we're looking at and like really represent the biology is tricky. And if you don't have that, you might fail. So yeah, make aging part of your drug discovery. Make sure that you're using an age system that's representative of your patient. You're going to get better outcomes. You can go after these like multimorbidity things. My nonprofit made a whole table of age-related diseases and how to go after them that you can use to sort of pick this, and then find some biomarkers. That's the end. Just to summarize the whole talk, aging is bad and it's very expensive. And if I was president, then I would definitely put a lot more money into targeting basic mechanisms of aging because you're going to get a dramatic ROI. It is solvable.
That's an important comment to make, your belief that it is solvable.
Yeah. I mean, this is all the way back to the beginning, right? But basically, we have organisms that live for hundreds of years. We have organisms that live for a few days. They share the same biology. And when we perturb biology that exists in both animals, like this insulin signaling pathway that you saw in the worms, it's roughly the same pathway in humans. And so there is no reason why this is not solvable. However, we don't know how hard it is, right? It is a complicated system. And so I am not going to tell you like, okay, in 20 years, like it's not engineering yet, basically, right? Like if we can't do a Manhattan project and just take these steps and it's solved. But we do know that like it has been solved in nature. It is doable. We should do it, not because it is easy, but because it is hard. As I said, we understand a lot, but we don't understand the whole system yet. We can do things regardless. And it's tricky to like fit this into our healthcare system. But if you do it right, and I think I don't even need to make this point anymore after GLP-1 drugs, right? Like it can be a whole different kind of biotechnology if we actually make things work here. We need to focus on just what are we able to measure and what are we able to use as prediction tools and so forth, right? It's not just like find the next target and the next treatment. The field needs to be fundamentally more capable, in my opinion. And there's no cavalry for this. There's no NASA. There's no, Calico didn't solve it, and Altos probably won't solve it either. Apologies again. We can't assume that someone is just going to solve this for us. And so if we want things to happen, we should get engaged. And of course, everyone in this room is already engaged, and I think that's amazing. But like, challenge can be solved, but we have to accept it.
All right, guys, let's give it up for Martin here.
Thank you.
So we now, for those of you who still want to eat anything, we have lunch outside. Martin will be there hosting Q&A at one of the larger tables. And we'll see you back here in 45 minutes.